Hello, and welcome to my video showing the different phases of meiosis. Today I have two helpers, Malin and Etta, my American Girl dolls. Are they here because I need an extra pair of hands? Yes. Are they also here because I just want to bring my American Girl doll collection into some schoolwork? Absolutely. But in all seriousness, let's get into what we came here for, the phases of meiosis. So first here we have the plate, which is going to function as our cell. For chromosomes, I have duct tape sticks that are tied together by a centromere, or in this case, a piece of ribbon. And also on these chromosomes, they have genes, the genetic coding. And with those genes come the alleles. And as you can see, there's different bands. On the pink ones, there's green and blue, and on the green ones, they have blue and pink. These bands stand for the genes and alleles. So this cell has 4 total chromosomes. Because this is the beginning of meiosis, we're in S phase, they are duplicated chromosomes. And this is a 2n equals 4 cell because there are 4 chromosomes and 2 pairs of homologous pairs. Homologous pairs are chromosomes that we get from our mom and dad. So in this picture, of the pink ones, one of them is from mom, one of them is from dad. In this case, we'll say that the pink one with the green bands is from mom, and the pink one with the blue bands is from dad. Now that we've established some of the basics of the tools that I'm using and what I'm talking about, let's go forward and jump into prophase. Meiosis has 2 goals. One is to produce gametes. That's the main goal. The second is to increase genetic diversity. And the first place that we see genetic diversity increase is in prophase. During prophase, the chromosomes layer on top of each other and mix up their genetic information a little bit. To make this simpler, I'm just going to use the pink chromosomes as an example. But what can happen while these chromosomes are entangling with one another? They can actually swap genetic information. So before we said that mom has the green and dad has the blue. Well, what can happen is when they untangle, mom can actually get a little bit from dad and dad can get a little bit from mom in these chromosomes. So as you can see in this picture, the alleles and genes look a little bit different because they've swapped. So now each of them has one blue and one green. This increase in genetic diversity is called crossing over. Also during prophase, 3 very important things happen. The first thing that happens is that the chromosomes condense. That was kind of already what we talked about when we said they were entangled. The second thing that happens is that the nuclear membrane disappears so that they can have a lot of space in the entire cell. And the third thing that happens is that the spindle forms. I will be using my doll's hands as spindle, which you will see in a moment. And that's the end of prophase 1. Now the cell is in metaphase 1. So the most important thing that happens during metaphase is that the chromosomes line up in the middle of the cell. It doesn't matter if it goes diagonally or from up and down or side to side, it doesn't matter, they just all have to line up. However, more importantly is that the chromosomes can line up in any order. So you could have the 2 pink next to each other or the green in between, and since there are 46 total chromosomes in each cell, that brings out a lot of diversity. So this is another place where we see an increase in genetic diversity. And this specific part here of the lining up of chromosomes is called independent assortment or random alignment. Now the cell moves on to anaphase 1. In anaphase, the spindle are going to grab the chromosomes by the centromere, and they are going to take the homologous pairs to separate sides of the cell. As this happens, the cell that we have now will start to pinch apart. And that brings us right to telophase, where we have the split chromosomes into 2 different cells. Cytokinesis often gets tagged on to telophase 1 because it is also the separating of the cell, but it's all the other stuff that isn't the chromosome, so like the mitochondria and that sort of thing. So now we have 2 new cells with 2 chromosomes in them, which means we have to go through meiosis too. Now these phases for meiosis 2 would be happening in both of the cells. However, I am just going to be showing one of the cells. Now going into prophase 2, the exact same things happen that happened in prophase 1. So the chromosomes will condense, the nuclear membrane will disappear, and the spindles are going to form. However, there will not be any more crossing over. That's only in prophase 1, not in prophase 2. Next, we have metaphase 2. And just like with metaphase 1, the chromosomes are going to line up on the cell end to end as we see here. Now in anaphase 2, the sister chromatids are going to split apart. So the 2 pink chromatids are going to separate and the 2 green chromatids are going to separate from one another. Then the spindle is going to grab 2 chromatids each and pull them to opposite sides of the cell. Again, the cell is going to start pinching at the top and bottom, wanting to separate into new cells. Now we are in telophase 2 and cytokinesis, and we have 4 daughter cells. These daughter cells are haploid cells, so all of them individually are n equals 2. In gametogenesis, if this is to produce sperm, there will in fact be 4 functioning cells of sperm. However, if this is for eggs, ovum, then there will only be 1 functioning egg and 3 polar bodies. Another thing to mention is that females will not go through the full process of meiosis until the egg has been fertilized, and then it can go into telophase. So that brings me to the third and final genetic diversifying that can happen during meiosis, and that is at fertilization, called chance meeting or random fertilization, when the sperm and the egg meet. Now the last bit of this video is going to be talking about the questions and the answers that I have and have had about meiosis. When I wrote the script for this lab, I had not heard of what non-disjunction was, but since writing the script, I have learned what it is, and it is when a cell goes through gametogenesis, and instead of having the chromosomes separate from each other, the homologous pairs during prophase 1, they get stuck to each other. And then what can happen is you end up with cells that have n+1 or n-1, so they have 3 chromosomes or 1 instead of the 2 at the end of meiosis 2. This is more common in females than males, and it especially happens in women over the age of 35 because their eggs are in them for so long that the chromosomes want to stick together. However, it is still possible for non-disjunction to occur in younger women or in males in their sperm cells. The second question that I had is what happens to polar bodies after they've been created? Like, where do they go? What happens to them? And one of my classmates actually asked this question in class today, and the answer is that they get shed with the lining of the uterus in the next menstrual cycle. Thank you so much for watching my meiosis lab, and I hope you enjoyed my demonstration of the process of meiosis and that you got a kick out of Malin and Etta. Anyway, I will be ending this video now, so bye! Thanks for watching!